So every atom of carbon and every calorie in your body comes from the process of photosynthesis. And the problem we have is that there's just not enough of it. The FAO predict that by 2050, we're going to need to get 70% more productivity out of our existing cropland. This is because of rising population, but it's also because of changing diets. And we're not improving productivity of crops at that speed. So basically, demand is rising faster than supply, which is why crop prices have doubled over the last 7 years, and we predict they're going to continue to double in real terms each 7 years as we go forward if we can't find new innovations to improve the rate of crop productivity. Now, photosynthesis is genetically highly conserved. It's very similar in soybean, rice, wheat, etc. Arabidopsis, a model plant, doesn't differ very much. There's very little genetic variation for breeders to use. But we do now have opportunities of engineering. And I've said too many times to my students, obviously, that photosynthesis is the final frontier for crop improvement, because a few weeks ago they took their revenge by posting this on my office door. So 70% more food by 2050. And clearly those price increases are catastrophic for the poorest communities in the world. From a corporate point of view, though, a 1% increase in yield of one of our major crops is worth a huge amount of money, 50 billion for soybean, for example. So what we did with NCSA's help now 10 years ago is really to simulate the process in silico. So each step of the process, there's over 100 of them, represented by a differential equation describing the underlying biochemistry or biophysics linked into a system which we could numerically integrate. And then, so we're able to simulate the process in silico. We can then apply optimization routines to it to say, well, our resource is optimally invested. And this is just a very subset of the data, and of course it depends on the conditions of the simulation, but what this basically showed to us was that it isn't optimized. And for example, we can pick out one enzyme in this example here, where the simulation is saying we need about 10-fold more of this particular enzyme, even though it's not a great deal of the total protein. Well, after we put out a number of these things in theory, the Gates Foundation stepped up and offered to put its money where our mouth was. Said, well, how about we pay you to test out some of these things? So what we're now doing is transforming plants with these changes, obviously the ones which the computer picks out as being best, and then testing those out. And this is that particular enzyme. We upregulated that in tobacco. You can see here the three plants on the right are the transformed plants. And we're now testing this in the field. So basically what we're doing is we're using this platform we call e-photosynthesis to predict changes we need. And these are both synthetic, changing the existing system, and system change, and synthetic, introducing new pathways. We then predict the change. The best of those we engineer into the plants. We phenotype that in controlled environments and then in replicated experiments in the field. One of our biggest limitations is around the data for photosynthesis, that despite photosynthesis being quite a conserved process, there's still the enzyme kinetic data and metabolite data are still difficult to actually obtain. There is an enzyme database which is pretty comprehensive called Brenda, another one called KEGG. Although it's not plants, Microbes Online is quite useful, and then literature. But really these databases are not at all well, they have the information in them, but extracting the information we need is extremely difficult from these, very time consuming. If we could reorganize all that data, This would make this process far more efficient and far more accurate. And then, of course, I know this isn't the problem of BrownDog, but we are now generating buckets of information, Illinois and our subcontractors, going all the way from sequence information right the way through to growth data of how these plants are growing every day, the weather, etc., around that. And anyone who can advise us on how we can do a better job here so that we don't present BrownDog with an even bigger problem in 5 years would be very helpful to us. Thank you.